going to be giving an overview of the next generation sequencing market, focusing primarily on the various platforms that are currently available. We're going to be looking at their strengths and weaknesses, as well as their suitability for a broad range of research applications that have been implemented on the next generation sequencing platforms. And as this is a very fast-moving field, we'll finish up with a discussion on where the market is heading and review some of the platforms and technologies that are on the horizon. So let me take a step back and let you know who we are and why we're in a good position to offer unbiased opinions on the next generation sequencing market. So BlueSEQ has set up an exchange where we match the needs of researchers with service providers and core labs who have the right technologies. Not only does this allow service providers to maximize the use in their machines, it ensures that researchers get timely access to the best, most appropriate next-gen sequencers, not just what their core lab happens to have to offer. And since this is done at a competitive bidding process, they can be assured of getting good value for their research budget. So BlueSEQ provides a platform for researchers and service providers to meet. Since we're neither a provider nor user of the technologies ourselves, we can offer unbiased opinions about the current market, instruments, applications, and so on, which is free information that can be found on the BlueSEQ Knowledge Bank on our website. So my apologies to those of you who have seen this graphic before, as it is such a compelling illustration of the impact next-generation sequencing has had on the cost of sequencing that it's been used in many talks. So using up-to-date data from the NHGRI, we're plotting the fully burdened cost of sequencing a human genome, with the most recent data representing about 30x coverage. Note that the y-axis here is plotted on a logarithmic scale, with each increment marking about a 10-fold drop in cost. And you can see the initial impact of the next-gen system from 454 right here in early 2008. And you could, as subsequent drops in price down to about the $10,000 range, these were driven by the release of more powerful systems from a handful of vendors. In fact, there's a few service providers in the range of $4,000 to $5,000 for research samples. And with more vendors entering the market, there's every reason to believe that the cost is going to be driven down to the somewhat arbitrary holy grail of $1,000, and even lower. So as much of this presentation will focus on the platforms that are currently on the market, I wanted to give an overview of the relative popularity of the various systems. I plotted from Nick Lohman's world map of high-throughput sequencers, which is a community-based effort to track next-generation system placements. This is just a breakdown of those system placements. While it's not a perfect measure of the market, it gives a pretty good estimation. And we can see that the field is really dominated by three main players. The first is 454 here, which was the first next-generation sequencing system on the market, and it has about a 17% market share. Subsequent systems from both Life Technologies at 19% and Illumina here at a commanding 60% were able to take the lead due to substantially lowering the costs. And so we're now going to go into more detail from each of the platforms. So Illumina is the current market leader with about 60% market share, and it has the broadest range of next-generation sequencing instruments. They use Illumina sequencing by synthesis chemistry, which is capable of generating high-quality reads up to 150 bases in length. For each slide, or for each of the platforms that we're talking about today, we're going to have a table similar to this one depicting the suitability of the instruments for a variety of common next-generation sequencing applications. Green is a good match, yellow is just okay, and red is an unsuitable match. The most common applications are human whole genome resequencing, exome sequencing, targeted sequencing, generally looking at a smaller region than the exome, transcriptome sequencing, where the goal is to look at things like transcript discovery, splice isoforms, novel 3' UTRs, etc., and RNA profiling, which is similar to transcriptome but more focused on comparing expression levels across multiple samples, and finally ChIP-seq here at the bottom. The HiSeq 2000 is Illumina's flagship platform, which offers our highest throughput at the lowest cost per gigabase. Each run occurring over 2 flow cells can generate over 1 billion reads and 600 gigabases of output, which translates into 5 full whole human genomes in 10 days. And internal runs at Illumina generate about a terabase of data at the moment. Despite having the lowest cost per gigabase, the machines are fairly expensive at a list price of $690,000. Also, a full run on the instrument costs about $20,000, and this we're talking about the HiSeq 2000. The HiSeq 1000 is a flow cell system similar to the 2000, but with half the capacity at a slightly lower cost. It's fully upgradable to the HiSeq 2000. Given the costs, both of the systems are best suited for those researchers who have a lot of experiments to run, which helps amortize the cost. Applications which require a lot of sequence per sample, like whole genome sequencing, exome, and transcriptome sequencing, are the best fit. The machines are certainly capable of running other applications, but there's a bit of a mismatch between what the machine can output and what is needed per sample. So in order to take full advantage of the low cost per base, many samples would have to be multiplexed together. Now in the middle column, the GA2X, based on earlier technology, this is a lower cost system at around $300,000 with a more modest output, generating about 95 gigabases per run. It can be a good choice for those with smaller budgets, but it's not based on the new HiSeq architecture, so it likely won't benefit from future HiSeq developments. It is, however, well suited for all of these applications. Moving one more to the right, the HiScan SQ with roughly half the output of the 2X is a hybrid system based on which can operate both as a sequencer and a microarray scanner, making this a good choice for those still transitioning from arrays to sequencing. Similar to the 2X, its output is a good match for most of the applications. And finally, in the right-hand column is the MiSeq, and that is the latest addition to the lineup. It is a so-called personal genome sequencer. Although it has a much lower output and higher cost per gigabase, the system is much less expensive at $125,000 per unit and is optimized for quick, low-cost runs. So, as an example, a full run of a paired-end 150 base read generating 1.5 gigabase of data takes only 27 hours and costs about $750. The output's far too low for the applications that really need deep sequencing like whole genome, exome, and transcriptome, as you can see in red. It's almost a good enough match for RNA profiling, but would only be able to run one sample at a time, and even then it's at the very low end of the accepted depth range. Certain ChIP-seq with small sample numbers might be a good fit, but what this machine is really built for is targeted sequencing, so if you're looking at a couple hundred genes across a few samples at a time. Since DNA libraries are compatible across various platforms with Illumina, some HiSeq owners plan on using the MiSeq as sort of a library prep QC. They can do a quick MiSeq run and then transition to the full run on the HiSeq. So moving on to Life Technologies, this is the number 2 competitor in the space with roughly 20% market share. Their solid platforms use a form of sequencing by ligation chemistry, which is able to generate high-quality reads of up to 75 bases in length. While some researchers feel this chemistry can provide higher quality data, it's pretty universally considered to be more cumbersome than sequencing by synthesis as it requires emulsion PCR. They have, however, announced that they're going to be releasing a non-emulsion PCR method called Wildfire in 2012. Life has 2 main systems based on their solid chemistry. The 5500XL is their flagship platform, which offers their highest throughput at their lowest cost per gigabase, with each run generating about 1.4 billion reads and 180 gigabase of output across 2 flow cells. The 5500 is a lower cost single flow cell system which generates half the output at 700 million reads and 90 gigabases per run. These are both high-powered systems with an application profile similar to that of Illumina's HiSeq. They're well matched for whole genome, exome, and transcriptome sequencing, and these are applications which benefit from a lot of sequence per sample. The other applications benefit when several samples are multiplexed together. And these systems have the advantage of being able to run individual lanes of a flow cell separately, making smaller projects more feasible. So 454 is the pioneer of next-generation sequencing, launching their first system in 2005. And while they've had a pretty healthy market share, or where they still have one at 17%, they've been surpassed by Illumina and Solid because of substantially lower cost per gigabase of these systems. The 454 systems use a form of pyrosequencing, which uses a cumbersome emulsion PCR, and this can struggle with homopolymers, or strings of the same base in a row, such as 6 A's in a row. However, it does have the distinct advantage of generating reads that are substantially longer than the other major platforms. The most recent FLX Plus kits are generating high-quality reads of up to 1,000 bases in length. Okay, so the GS-FLX here on the left-hand column, with about 1 million reads and a gigabase of output, is best suited for the lower output applications like targeted sequencing. And for the remainder of the platforms that we'll be talking about here, we've swapped out human whole genome for small genomes, and so these are things like bacteria or viruses. The long reads of the 454 are especially suitable for de novo small genome assembly. And the long reads are also beneficial for discovering transcript structure. Since it doesn't have high enough counts or reads per run, the transcriptome sequencing, for transcriptome sequencing, is often combined with one of the high count systems from Illumina or Life Technologies. The GS Junior is a less powerful system than the FLX, offering about 1/10 the output, but at 1/4 of the instrument cost at about $100,000. This has an average read length of 400 bases in a 10-hour run, and it's geared for researchers who have a more modest sequencing need. Okay, so moving on, the Ion Torrent. This is now a subsidiary of Life Technologies, and it's the newest entrant into the next generation sequencing market. It currently has a market share of 3%, but rapid improvements to the platform in short runs of about 2 hours are causing it to gain popularity. It uses a pyrosequencing chemistry similar to 454, but instead of using fluorescent dyes, the system measures the release of hydrogen atoms or protons, meaning it essentially acts as a big pH meter. Like the 454, the system is capable of producing longer reads than the short read technologies. And as of recently, it was producing high-quality reads of about 150 bases, and they've released a new kit that takes the reads beyond 200 bases. However, it still suffers from the pyrosequencing Achilles heel, difficulty dealing with homopolymers. While the Ion Torrent doesn't have multiple machines, it does offer multiple chips, each with different capabilities. The 314 and 316 are the two officially commercially released chips with official output specs at 10 megabases and 100 megabases respectively. Real-world experiments have shown substantially higher outputs though. The latest runs from Life have generated over 500 megabases on the 316 chip. The 318, which is made for 1 gigabase runs, is still in the testing phase and plans for commercial release by the end of the year. Despite the rapid improvements, the relatively low output and small number of reads limit these chips to the lower output applications like targeted sequencing. The 316 and 318 are also a good fit for small genome projects, and while there's an interest in using this technology for RNA profiling and ChIP-seq, they just generate enough reads to make these applications truly feasible. Okay, so Pacific Biosciences is interesting in that they're the only company with a single molecule sequencer on the market, the PacBio RS. Helicos actually got there first with the Heloscope, but they're no longer selling the instruments, and they're only operating as a service business. The PacBio RS is also unique in that it's able to generate the longest reads by far, with an average greater than 1,500 kilobases, and individual reads in the 5 to 10 kb range. Despite these unique advantages, PacBio only has about a 1% market share, and this is likely due to the cost, over $700,000 for the instrument, and it also has limited output, which is about 40 megabases per 40-minute run. So the small output limits the applications the RS can be used for, with small genome and targeted sequencing being the only two good fits there in green. The long reads would be especially useful for de novo transcriptome work, but the extremely limited number of reads per run, currently at about 35,000, puts this application out of reach. However, there's quite a bit of interest in combining the long reads of the RS as a scaffold for the reads of other higher throughput systems. The other potential advantage of the RS is the possibility to directly measure modified bases, such as the 5-methylcytosine, without the use of bisulfite conversion. However, this capability is still quite a long way from being reduced to practice. Okay, so Intelligent Biosystems, Asco Biotech, and Dover have joined forces to release the MaxSeq instrument. IBS provides the chemistry, Dover provides the instrumentation, and Asco provides the marketing and service support. It uses a sequencing by synthesis chemistry and is essentially a cheaper version of Illumina's GA2X, both in terms of instrumentation and operating costs. With an output of up to 135 gigabases and 1 billion reads per run, it's a pretty decent match for most high-throughput applications. Complete Genomics is unique in that they have their own proprietary sequencing technology. They offer it only as a service. They use a somewhat complicated form of sequencing by hybridization, which produces non-contiguous 35 base pair reads. They've also decided to focus just on one application, human whole genome resequencing. They're competing heavily with the likes of Illumina's Genome Network and BGI, and are currently offering whole human genomes at currently $4,000 to $5,000. They haven't made any indication of whether they're going to be expanding their services into other areas in the near future. So that covers it for the major next-generation sequencing platforms that are currently on the market. I'd like to spend the last few minutes to review the trends that we're seeing going forward and have a peek into the technologies that are currently in development. With the current players being very well established, new entrants are attempting to develop platforms which address niche markets or take substantially different technical approaches, or both. The most common approach is the use of nanopores, where individual DNA molecules are threaded through microscopic pores. These pores are either modifications of naturally occurring protein pores or solid-state pores constructed from semiconductor methods. So all of the upcoming systems are attempting to perform faster reads, reducing the DNA to data time from days down to a couple of hours or minutes. And this fits well with the strong push in the diagnostic space, especially for point-of-care diagnostics. The new systems are also being differentiated on the amount of output they generate. While some systems are pushing for more and more output, others are sacrificing output for the sake of speed. So perhaps one of the most anticipated systems is the GridION from Oxford Nanopore Technologies. As the name would imply, the sequencing occurs via nanopore. While they have a few projects in the work, their first commercially available system will likely be exonuclease sequencing via the alpha hemolysin nanopores. The system is a bit different from the more traditional nanopore sequencing in that instead of threading an entire DNA strand through the pore, individual bases are cleaved off by an exonuclease and they migrate through the pore one at a time. Sequencing is performed using disposable cartridges, which you can see here, and they can handle up to 96 samples at a time. The cartridges are fed into the GridION unit, which is the which handles sample distribution and basic data analysis. And as one of the most intriguing aspects of the GridION system is that several of these units can be linked together just like computer blades to form a powerful sequencing cluster. While specific dates haven't been released by the company, it is thought that the instrument might start hitting the field by the end of the year with a commercial release sometime in 2012. NobleGen is another example of a single molecule sequencer which would use nanopores for the sequencing readout. But rather than sequencing native DNA, they convert the DNA into expanded synthetic representations, which are longer strings of bases that can hybridize with color-coded beacons, one for each of the 4 bases. As the modified DNA molecule is threaded through the nanopore, the beacons are stripped off, briefly emitting a flash of color, which is recorded and translated in sequence. Because of the conversion process, the initial reads are limited to about 200 bases. NobleGen is targeting the diagnostic market and anticipates a commercial launch in 2014. Stratos Genomics is also taking a nanopore approach and is also converting the original DNA molecule into an expanded molecule, or what they call an expandomer, and this is easier to sequence. The expandomer is created through the incorporation of X probes, which are small oligos complementary to the target sequence, which have reporter molecules attached. When the newly synthesized strand containing the X probes is expanded, the synthetic molecule is about 50 times the size of the original DNA molecule. The expandomer is then threaded through a nanopore, which reads off the sequence as represented by the reporter molecules. Nabsys is creating yet another nanopore-based sequencer. However, rather than directly measuring individual bases or modified molecules, the system uses a sequencing by hybridization approach. Individual strands of DNA are fragmented into roughly 100 kb pieces. So these fragments are roughly 100 times larger than the fragments by the short-read technologies. And these fragments are then hybridized to a particular hexamer probe, which will hybridize at several locations along the 100 kb strand. This partially hybridized 100 kb sequence is then threaded through the nanopore, which can measure via electrical conductance changes where the hybridization events have occurred. Hybridization patterns from other 100 kb strands with the same hexamer are then combined to create a genome-wide probe map. This process is repeated in parallel with all possible hexamers, and the resulting probe maps are then combined to generate a complete sequence of a genome. So 454 has partnered with IBM to create a DNA transistor-based nanopore sequencer. The system will use alternating layers of metal and dielectric material to control the rate of passage through the nanopore, which is one of the biggest challenges of nanopore sequencing. This technology is still in the early stages, but many are hoping that the combined resources of IBM and 454 Roche will be able to provide a more powerful system to the market. So the last system we'll talk about today is from NuBio, and this is a $50,000 machine which is currently in beta testing. It uses a form of sequencing by hybridization which occurs in picoliter drops. Rather than attempting to outmuscle the market leaders, NuBio is focusing on inexpensive $30 to $200 quick runs on targeted amplicons, so up to 100 genes of 1 kb each. This is a combination of features they hope will be very attractive to the diagnostic market. I know I went through a lot of numbers pretty quick, and so I would encourage you to visit our knowledge bank on our website here at the address at the top. If you have any more questions, feel free to send me an email or my colleague Sean Baker. And otherwise, just please take a look at our website, and you can find the most up-to-date next-generation sequencing information here at our knowledge bank on our website. Thank you for joining me today.